Bismillahirrahmanirrahim. Assalamu alaikum wa rahmatullah. আমরা আজকে রিভিউ ক্লাস অন জেনারেল ব্যাকটেরিওলজি আমরা তো দুটো ক্লাস নেব। এই ক্লাসগুলো পরীক্ষার জন্য খুব ইম্পর্টেন্ট। আমরা সাধারণত ময়মনসিংহ মেডিকেল কলেজে প্রফেশনাল পরীক্ষার আগে আগে এই ক্লাসগুলো নিতাম এবং এগুলো ভাইভা এবং রিটেন দুটোর জন্য খুব উপকারী। Review class on General Bacteriology Part 1. আমরা সরাসরি যে সমস্ত প্রশ্নগুলো জিজ্ঞেস করা হয় বা যেগুলো সংক্ষেপে খুব গুরুত্বপূর্ণ সব মনে রাখতে হয় সেগুলোই এখানে আলোচনা করি। প্রথমে আমরা শুরু করি। Microbiology deals with the infectious agents, the disease agents. So how can you classify the disease agents? State the differences among bacteria, virus, and fungi. We know the disease agents are bacteria, protozoa, helminths, fungi, virus, and prions also. Bacteria, they belong to prokaryotic kingdom. Fungi and protozoa, they are protist kingdom. Helminths, they belong to animal kingdom. Viruses are non-cellular, and there is no pathogen in the plant kingdom. What are the points of differences among these agents? We consider the cell, the nucleic acid content, nucleic acid type, ribosome, mitochondria, their motility, replication, outer coat, etc. This is the common question: what is prokaryotes and eukaryotes? What is the basic difference between them? What are the other differences? You see, the literal meaning of prokaryotes means pro means primitive and karyotes means nucleus. So cells with primitive nucleus are prokaryotes and cells with advanced nucleus are eukaryotes. Basic difference is eukaryotes have nucleus with a nuclear membrane, whereas prokaryotes do not have true nuclei with nuclear membrane. Rather, they have nucleoid. Other differences include number of chromosome, nucleoli, mitochondria, Golgi complex, and endoplasmic reticula. These are the prokaryotic cell morphology. You see, these are large pictures, electron scanning picture. You see, these are coccus, bacillus, vibrio with curved, and there are some straight coccobacilli are there, spirochetes are there, spirillum are there. So all these, and also there is possibility of pleomorphic bacteria. And how they group, based on their grouping, they can be classified as diplococcus or the chains of cocci. Sometimes they remain in irregular clusters. And this is due to their binary division in single plane or different planes. And they help in identification of the bacteria in Gram-Stein smear. Another important question is, how can you classify bacteria? And bacteria can be classified in many ways, but the clinically important and easiest classification is this. Say, we can classify a rigid, thick-walled bacteria, flexible, thin-walled bacteria, and wall-less bacteria. So wall-less bacteria, you must remember the name Mycoplasma. Flexible, thin-walled bacteria, Treponema, Borrelia, and Leptospira. A rigid, thick-walled bacteria, These are the common bacteria. They are free-living extracellular. They include gram-positive cocci and bacilli, gram-negative cocci and bacilli, and acid-fast bacilli. So this is the simplest classification of bacteria. And this free-living bacteria, the common bacteria, if we elaborate them, gram-positive cocci may be Staphylococci and Streptococci based on their arrangement. Bacilli, again, spore-forming, non-spore forming. Spore-forming bacilli are aerobic bacillus, anaerobic Clostridium. Non-spore forming bacillus are non-filamentous, like Corynebacterium and Listeria. Filamentous, Actinomyces nocardia. Gram-negative bacteria also can be cocci and bacilli. Cocci, Neisseria cocci, bacilli may be aerobic, anaerobic, and facultative. Facultative may be straight and carved, straight, respiratory, zoonotic, enteric, and gram-negative aerobic, and there are anaerobic cocci also. This is another question. What are the essential and non-essential structure of bacterial cell? Essential structure only 5, only 4 for all, and fifth one is common to, fifth one is for gram-negative bacteria. So cell wall, most important, cell membrane, nucleoid, cytoplasm, these are common. Of course, there are some exceptions. There are some cell wall-less bacteria like Mycoplasma. Periplasmic space, this is the essential structure for gram-negative bacteria. It is absent in gram-positive bacteria. So if you write that periplasmic space, you will have to mention it is for gram-negative bacteria only. What are the non-essential structures of bacterial cell? All other structures like flagella, fimbria, capsule or cilia, spore, plasmid, transposon, these are all non-essential structures. Without these structures, bacteria can survive. What are the functions of essential and non-essential? Essential structures are required for their survival. Non-essential structures mostly act as virulence factors. What is the basic structure of cell wall? You see, cell wall is unique in case of bacteria, and basic structure of cell wall, they have got a important component, peptidoglycan. This peptidoglycan composed of alternate strands of 2 major subunits, N-acetyl glucosamine, N-acetyl muramic acid. These 2 subunits, they are cross-linked by tetrapeptide chain attached to the N-acetyl muramic acid. Interpeptide bridge occurs in gram-positive cell wall. In gram-negative cell wall, in addition, there are lipoprotein layer, outer membrane layer, and lipopolysaccharide layer. You know, this name is why peptidoglycan. There is peptide as well as glycan, N-acetyl glucosamine and N-acetyl muramic acid. So its name is peptidoglycan. And you see, this is the picture of a diagrammatic picture of a gram-positive bacteria. There is a thick layer of peptidoglycan, very thick, 1, 2, 3, many layers. And these are, there is N-acetyl glucosamine, N-acetyl muramic acid, teichoic acid, they are cross-linked, and it is gram-positive. How gram-negative bacterial cell wall differs from that of gram-positive bacteria. First of all, the cell wall of gram-negative bacteria is much more complex. The peptidoglycan layer is very thin, 1 or 2 layers. And there is additional layer filled and surrounded with additional layer of periplasmic space, which is protein-rich gel-like fluid is there. They have just between the inner membrane and outer membrane. There is unique outer membrane on top of the gram-negative bacteria, and there is lipopolysaccharide that acts as endotoxin, and lipid and O-specific polysaccharide, which helps in antigenicity. What are the functions and clinical significance of cell envelope? Cell envelope consists of cell membrane, cell wall, and all other things. The first function is they maintain shape and size, cocci, bacilli, etc. Protects from osmotic lysis. And clinical significance, these are the major determinants of virulence in both groups of bacteria, gram-positive as well as gram-negative. Endotoxin, which is inherent to all gram-negative bacteria, is toxic to animals in a variety of ways. Peptidoglycan and lipoprotein, as well as some teichoic acids, they can induce the alternate complement pathway, leading to inflammation and lysis. Teichoic acids and O-specific polysaccharides may be used as adhesive. What is the functions of clinical significance of cell wall? Some cell wall components protect against phagocytic engulfment or digestion, like capsule. Variations in the macromolecular structure of cell wall is the basis of antigenic variation. And outer membrane of gram-negative bacteria is target for complement, hydrophobic agents, and certain antibiotics. And peptidoglycan layer is dismantled by a host enzyme known as lysozyme. Many antibiotics are specifically directed at peptidoglycan synthesis. So these are all clinical significance. For example, penicillin, cephalosporin, they work by damaging the pentaglycine cross bridges of peptidoglycan. What is the peculiarity of cell wall of acid-fast bacteria? This group of bacteria have unusual cell walls and thereby they resist decolorization. They contain high concentration of lipids called the mycolic acid, and from this came the name mycobacteria. The mycobacterial cell envelope is extremely hydrophobic and forms an exceptionally strong permeability barrier, rendering the mycobacteria naturally resistant to a wide variety of antimicrobial agents. Therefore, there are some special anti-TB drugs. Other antibiotics do not act on mycobacteria. And this is the diagram of cell wall of acid-fast bacteria. You see, in addition to peptidoglycan, there is arabinogalactan and mycolic acid layer. And these mycolic acid are very hydrophobic. They resist decolorization by alcohol and to certain percentage of acidos. Cell membrane. Cell membrane, bacterial cell membrane and human cell membrane, they have some similarity. There are two structural components, double layer of phospholipids, you see, and protein layer, and a fluid mosaic model. The bacterial cell membrane consists of saturated or monounsaturated fatty acids, never polyunsaturated fatty acids, and they do not contain normally sterols. By this way, it differs from eukaryotic cell membrane. Cell membrane, they completely encloses the bacterial cell, that is protoplast. They're composed of 60% protein and 40% phospholipid and arranged as a bilayer. And what is the function? Their main function is active transport of molecules into the cell, energy generation by oxidative phosphorylation, synthesis of precursor of cell wall, and secretion of toxins and enzymes. There is a special region in the cell membrane known as mesosome. This is an invagination of cell membrane and important during cell division. They function as origin of transverse septum and they divide the cell into half. And this mesosome is binding site of DNA also. Ribosome, this is also another essential structure. Bacterial ribosomes are the site of protein synthesis. They differ from eukaryotic cells by size and chemical composition. Bacterial ribosomes are 70S in size with 50 and 30S subunits. Human ribosomes are 80S in size with 60S and 40S subunits. S means 7-part unit. This difference is the basis of selective toxicity for many antibiotics. Let us discuss about the nucleoid. This is unique to bacteria. Nucleoid is the area of cytoplasm where DNA is located. It is called nucleoid because it doesn't have any nuclear membrane. It's a single circular molecule with a molecular weight of 2 to the power 10 to the power 9 and contains about 200 genes. Human contains about 100,000 genes. Bacterial nucleoid has no nuclear membrane, no nucleolus, no mitotic spindle, no histone, and no intron. There are many no. No nuclear membrane, no nucleolus, no mitotic spindle, no histone, and no introns. Capsule, this is a non-essential structure, but it is a very important virulence factor for many bacteria. Capsule is a gelatinous layer covering the entire bacterium, composed of mostly polysaccharide except Bacillus anthracis, which contains glutamic acid. There are many types of capsule. Capsule, slim layer, glycocalyx. If capsule is organized and firmly attached to the cell wall, this is called capsule. If it is unorganized and loosely attached to the cell wall, this is called slim layer. And if it is a thin polysaccharide coating that is secreted by many bacteria, it covers like a film and helps in adherence to various surfaces. Functions and clinical significance of capsules. First of all, it contributes to the virulence of bacteria by preventing phagocytosis, by WBCs, and prevents attack by antimicrobials. And capsulated bacteria, they need to be opsonized by presence of antibody against the capsule. It prevents drying out or desiccation. It allows the bacteria to adhere to various surfaces, and it helps in identification of bacteria by using antisera like pneumococcus, meningococcus. There are many types. And capsular swelling test or coiling reaction helps in diagnosis of the capsulated bacteria, and they help in typing of bacteria on the basis of antigenicity of the capsule. And there another important thing is they are used in vaccine preparation. Pneumococcal, meningococcal vaccine are prepared from their capsules. What is the structure of a flagella? Flagella is composed of many subunits of single protein flagellin. Flagellin arranged in several intertwined chain, and it has a filament, a hook, and basal body. Filament, hook, and basal body. And this is the detailed structure. You see the basal body, it encodes in the cell, in the bacteria, the outer membrane, the cell wall and cell membrane. Its origin is from cytoplasm. And the movement in a nice way, you see. How they can arranged. The flagella can be arranged of different types based on their arrangement. They can be monotrichous on this one flagella. Vibrio is a classical example. Lophotrichous, tuft at one end, amphitrichous, both ends, and peritrichous all around the bacteria. Peritrichous, classical example is Enterobacter C. Amphitrichous, Lophotrichous, Pseudomonas, they are the examples, and monotrichous Vibrio. What is the clinical significance of flagella? Flagella are important for motility and virulence in case of Vibrio cholerae, Escherichia coli, Proteus. And they are also required for antigenic determinant. They interfere with phagocytosis, helps in identification and typing by antibody detection. Also helps in diagnosis of disease by antibody detection, like in Vidal test. Vidal test, the flagellar antibody, that is TH, AH, and BH, are very important. They can be, there are some endoflagella as well. Spirochetes move by using a flagellum-like structure called axial filament, which wraps around spiral-shaped cell to produce an undulating motion. অনেকে চাদর গায়ে শীতকালে যারা চাদর গায়ে দিয়ে থেকে হাত ভিতরে থাকলে যেরকম হয় সেটা হলো এন্ডোফ্লাজেলার এক্সাম্পল। Pili. Pili are hair-like filaments that extend from cell surface. They are shorter and straighter than flagella, and they are composed of protein known as pilin. They are found mainly in the gram-negative bacteria, and there are different types of pili or fimbria, and a special type of pillar, that is F pillars, or known as sex pillars, mediates transfer of DNA between mating bacteria. What are the functions of common pili? Pili plays role in adhesion of the bacteria to specific receptors in the host cell, exchange of genetic material by a specialized kind of pili, that is known as conjugation. They also helps in avoidance of immune response, and antigenicity is there. So pili of different bacteria are antigenically distinct, and they are used for antibody detection test. Antibody of one bacteria cannot prevent the attachment of another species, and there are some species they have got some antigenic variation. To be proud of learning is the greatest ignorance, and we should remember these things. Bacterial endospores. Bacterial endospores, they are metabolically inactive. They are produced in response to nutrient limitation or extreme environments. They are highly resistant. They are stable for years, not reproductive, and only some special bacteria like Bacillus and Clostridium, they can sporulate. So these are the, and what is the functions or clinical significance of spore? Because medical microbiologists who do not study anything about microbe which is not, which doesn't have any clinical significance. So this is an important question. What is the function or clinical significance of a spore? Why spores are so tough? And some examples, of course. The spores are highly resistant due to some following features. First of all, it has got thick coat like core, cortex, coat, and exporium. আমরা একটা এক্সাম্পল দেই নারিকেলের সঙ্গে, নারিকেল যেরকম বাইরে থেকে ভিতরে অনেকগুলো লেয়ার আছে সেরকম। It is in dehydrated state, metabolically inert. It has got keratin-like protein in the coat, the নারিকেলের ওই খোসার মতো, and calcium DP-cholic acid is present in the cortex, and finally DNA repair enzymes contained within the endospore and able to repair damaged DNA during germination. Very important. So clinical significance is difficulty in sterilization. They need autoclaving. They cause some dreadful diseases like tetanus, gas gangrene. The difficulty in diagnosis for spore-bearing bacteria, and sometimes they can be used as biological weapon, that is anthrax. So examples, also know, there are aerobic and anaerobic bacillus like Clostridium tetani, perfringens, Clostridium botulinum, and aerobic is Bacillus anthracis. Plasmid. Plasmid is an extra chromosomal DNA molecule. This is unique to the bacteria. They encode from 40 to 50 zones. They contain useful but not essential. And plasmids, they contain many virulence factors, including the resistance genes. Most common mode of transfer is conjugation, and they determine the important characteristics such as production of toxin, enzyme, etc. There are two types of plasmid, conjugative and non-conjugative. Conjugative plasmids are transferred from bacterium to bacterium, usually members of the same species or very closely related species through conjugation. Non-conjugative plasmids, common in gram-positive cocci as well as in some gram-negative organisms like Haemophilus influenzae, Neisseria gonorrhoeae, etc. What are the mechanism of action of antibiotics? Antibiotics, you know, there are targets are cell wall, cell membrane, protein synthesis, and nucleic acid from outside to inside. So many bacteria like penicillin, cephalosporin, vancomycin, they act by inhibiting cell wall synthesis. Polymyxin, pollen, they act on cell membrane with impairment of permeability. Some bacteria, they act on, some antibiotics act on protein synthesis like aminoglycosides, macrolides, tetracycline, chloramphenicol, and some act on nucleic acid synthesis inhibit like quinolone, rifampicin, metronidazole, etc. Microbes can be classified on the basis of oxygen requirement. There are following groups. One group is obligate aerobe. The oxygen is must for their growth. Another group is obligate anaerobe. They cannot grow in presence of oxygen. They grow only in absence of oxygen. In between is facultative anaerobe. They grow with or without oxygen. There are some aerotolerant anaerobe grows in presence of little amount of oxygen, but they does not use it. They are not killed by the oxygen, but they do not use it. Microaerophilic, they grows in low oxygen concentration. So you can see the obligate aerobe, they will grow where oxygen is present on the surface of the media. Obligate anaerobe on the deep where there is no oxygen. Facultative anaerobe throughout the media. Aerotolerant also, and microaerophilic. Why is oxygen harmful to some bacteria? Oxygen is a very strong oxidizing agent. It pulls electrons off other molecules. And when oxygen is utilized, singlet oxygen, superoxide free radical, hydrogen peroxide, and hydroxy radicals are formed. These compounds are very toxic, and cell is harmed if it cannot get rid of toxic forms of oxygen. To get rid of the toxic oxygen compounds, cells, they need two enzymes, superoxide dismutase and catalase. If some bacteria do not have this superoxide dismutase and catalase, they are killed by the toxic form of the oxygen. What is sterilization, disinfection, and antisepsis? Sterilization is a process by which an object is freed from all viable organisms, including spores. This is an absolute state. Disinfection is a process of destruction of vegetative form of pathogenic organism capable of producing infection. And antisepsis is a process that applies to prevent infection in living surface. So disinfection and antisepsis, there is difference. Disinfection are toxic, they cannot be used on animal surfaces. but antisepsis can be used in living surfaces. What are the methods of sterilization and what is the principle of autoclaving? Very often it is asked in the Viva and in the written also. Methods of sterilization are heat, ionizing radiation, filtration, sterilizing gases, liquids. There are plasma sterilization also. Principle of autoclaving: steam saturated at a high pressure and temperature is a better sterilizing agent than the dry heat. This is known. And bacteria are intrinsically more susceptible to moist heat as bacterial protein coagulates rapidly in moist heat. This saturated steam heats the article rapidly by releasing latent heat, which kills the bacteria. And steam contracts in volume, that is 1600 ml of steam. Steam at 100 degrees centigrade condenses into 1 cc water, and thereby it enhances penetration. So autoclaving is most efficient process of sterilization due to following reasons. So what are the reasons? Moist heat kill the organisms and spores at lower temperature and in shorter duration, because water helps in disruption of non-covalent bonds, that is hydrogen bonds, and thereby disrupt secondary and tertiary structure protein. Say, this is an example. If we want to kill a bacteria at 100 degrees centigrade by dry heat, it requires 20 hours. Whereas at 120 degrees centigrade by moist heat, only 15 minutes is required. Second point, penetrating power of moist heat is better than dry heat due to low density of steam. Condensation of steam and contraction of its volume creates a negative pressure on the surface. This is 1600 ml of steam condenses into 1 ml of water, and liberation of latent heat from 1 ml of water, 518 calories are liberated. So liquid or watery solution or articles retain their structure in moist heat but not in dry heat. Some chemical sterilizing or sporicidal agent, you should know chemical. There are gaseous and liquid forms. Gaseous are ethylene oxide, glutaraldehyde, formaldehyde, hydrogen peroxide. Hydrogen peroxide is now popular in many sophisticated laboratories. Ethylene oxide is also used. Among the liquids, the formaldehyde is a classical one. Glutaraldehyde now it is used very much, and very cheap one is chlorine releasing agent, that is sodium hypochlorite, popularly known as bleaching powder. Iodine and iodophores are also used. What is an antibiotic and what is the basic principles of antimicrobial therapy? Antibiotic is a substance produced by another microorganism, naturally or synthetically, that inhibits the growth of other microorganisms. What is the basic principle? It must have selective toxicity. That is, it's selectively toxic to the organism but not the host. And it should reach the site of infection at inhibitory concentration. So in vitro it is selectively toxic, but if it cannot reach the site of infection, it will not act. Penetrate and bind to target, avoiding inactivation and exclusion. These are the basic principles for an efficient antibiotic. How can you classify antimicrobials? They can be classified on different bases: by origin, by their mode of action, by target site, and chemical structure. By origin, they may be natural antibiotics, natural products of fungi and bacteria. Synthetic compound, trimethoprim. They are either bactericidal or bacteriostatic. Bactericidal, they kill the organism directly, and bacteriostatic, They prevent organism from multiplying, and host defenses then do the rest. By target site, cell wall synthesis, cell membrane function, protein synthesis, inhibition, nucleic acid synthesis, and by chemical. So, which organisms frequently develop resistance? We should know it. Staphylococcus aureus, Escherichia coli, Klebsiella, Enterococcus, Pseudomonas, Mycobacterium tuberculosis. So Staphylococcus, when they are resistant, they are known as MRSA and VISA. Escherichia coli, Klebsiella, when they are resistant, ESBL, extended spectrum beta-lactamase producer. Pseudomonas aeruginosa, they are intrinsically resistant to many antibiotics, and also their Mycobacterium tuberculosis is also very resistant to many antibiotics. They are MDR and XDR TB, multi-drug resistant. How microorganisms develop resistance? Microorganisms, they develop resistance by four ways: by producing some drug-destroying enzyme like penicillinase, beta-lactamase, change in the permeability to the drug, for example, tetracycline and Pseudomonas aeruginosa, change in the receptors, which decrease antibiotic binding and action, change in protein binding, penicillin binding protein and amino acid, altered metabolic pathway, sulfonamide resistant bacteria utilize preformed folic acid and do not require the presence of power. How can you delay the emergence of drug resistance? You should prescribe only when it is needed. You should not prescribe antibiotic in viral infection. And when you prescribe, it must prescribe at adequate dose and duration. And narrow spectrum antibiotic is preferable. And you must limit the use of newer drugs. And sometimes you need combination of drugs in special cases. And minimize use of antibiotics in the livestock. How drug resistance develops? There are two ways: non-genetic origin and genetic origin. Non-genetic origin, genetic origin, there are chromosomal and extrachromosomal. Chromosomal due to mutation like MRSA, MTB, and extrachromosomal, there is plasmid mediated, say for example, penicillin resistance. And non-genetic origin, there are sometimes the bacteria can be metabolically inactive. Sometimes the specific target structure can be lost, like cell wall, if cell wall is lost. And sometimes sites where antimicrobials are excluded or are not active, say sometimes salmonella are intracellular and antibiotic cannot reach there, like these aminoglycosides do not enter the cells. These are the non-genetic or origin of drug resistance. When combination of drugs are used, combination of drugs are used in severe infection, mixed infection, prevention of resistance or delaying of resistance in case of tuberculosis, decreased toxicity, and enhanced bacterial action. How a bacteria cause disease and what are the virulence factors of bacteria? Bacteria cause disease by different virulence factors, also known as determinants of pathogenicity. These are, they can adhere by pili or colonizing factor antigen, damage of mucosal defense by IgA protease. Some bacteria can interfere with phagocytosis by capsule. Some bacteria have invasive properties. They can enter through the cell barrier inside the cell. Some bacteria have capability of intracellular multiplication. Some of the bacteria, they can secrete toxins and enzymes. What are the biological effects of toxin? Biological effects of toxin include fever due to release of interleukin-1 and hypotension, shock, impaired perfusion of internal organs due to release of tremor necrosis factor and nitrous oxide from activated macrophages, which causes vasodilation. DIC, disseminated intravascular coagulation, and inflammation due to activation of alternate pathway. What are the different types of media? Media can be classified based on the composition and based on consistency. Based on composition, synthetic and complex media. Based on consistency, solid, liquid, or soluble. The synthetic media contains a specific amount of ingredient, but complex media contains just some ingredients which are not specifically different components, but mixed. It can be different into basal media like nutrient agar media, contain the basic nutrients for all bacteria. Selective media, vacuum ki agar. Differential media, again, vacuum ki agar. Enriched media, blood agar, chocolate agar, etc. And enriched media, that is also selective. What is the clinical importance of normal flora or residence flora? Opportunistic infections, normal flora in unusual site, they can cause opportunistic infection. For example, bacteroids from intestine, when go to the deeper tissue, they result of opportunistic infection. Staphylococci from skin and nose, if they go to other side, they cause infection. And Candida causes pneumonia in people undergoing cancer therapy or immunosuppression. Pneumocystis caronae, common inhabitant of the lung, causes pneumonia and death in AIDS patient. What is the beneficial effects of normal flora? Normal flora, they keep out the invaders. That is, they interfere with the adherence of pathogenic bacteria. Antigenic stimulation by normal flora, they serve as a defense mechanism. Lower concentration of bacterial stimulation leads to production of IgA, which is secreted through mucous membrane. And they have some role in nutrition and metabolism. E. coli and Bacteriology, they synthesize vitamin K. What are the sterile sites of the body and what is its clinical importance? Sterile sites in the body include blood, CSF, synovial fluid, and deep juice. If some pathogenic bacteria in this site are found even in very low concentration, this indicates a disease. What is generation time and what is growth curve? Generation time is average time required for a bacteria to divide. That is also known as doubling time. What are the requirements for bacterial growth? Bacteria, for their growth, require physical and chemical factors. Among the physicals are temperature, pH, and osmotic pressure. Chemicals are carbon, nitrogen, sulfur, phosphorus, trace elements, oxygen. Oxygen is both useful and harmful. Organic growth factors like vitamin, amino acid, purine, and pyrimidines. Significance of phases of growth curve. You know, there are 4 phases: lag phase, log or exponential phase, stationary phase, and phase of decline or death phase. Lag phase is the time required for adaptation. During this time, enzymes and intermediates are formed and accumulate until they are present in optimum concentration. In exponential phase, they grow at a exponential, and this growth occurs until 2 things happen. Either one or more nutrients in the medium become exhausted, or toxic metabolic products accumulate and inhibit growth. Most of the antimicrobials act efficiently in this phase. A stationary phase, eventually exhaustion of nutrients or accumulation of toxic product causes the growth to cease, and there is slow loss of cells through death, which is just balanced by the formation of new cells. Toxins and spores are formed at this phase. And phase of decline, death rate increases until it reaches a steady level. And only persisters remain for months or even years. Sometimes it is asked about the agar. This is a solidifying agent. This is a complex polysaccharide. It is used as solidifying agent for culture media, and they are generally not metabolized by microbes. This unique property liquefies at 100 degrees centigrade but solidifies at 40 degrees centigrade. What is Koch's postulate and what are its components when it cannot be proved? The postulate of Robert Koch that establishes the etiological relationship of microbe with a disease. First component is bacterium found in all patients having the disease. Second component, from that diseased patient, bacterium should be isolated and grown in pure culture. Third component, from this pure culture should be inoculated in susceptible animal and produce disease. And fourth, from the experimental animal re-isolated in pure culture, and this is, if these 4 points are fulfilled, then Koch's postulate is fulfilled. Some microbes cannot be cultured in artificial media like Treponema pallida, Mycobacterium lepti. Some do not have, some cannot produce in experimental animals. In those cases, Koch's postulate cannot be proved. What is the clinical significance of periplasmic space? This is an important question. Periplasmic space present only in gram-negative bacteria. Some binding protein for transport of nutrients like amino acid, sugar, vitamin, ion remain in this space. There are some hydrolytic enzymes like alkaline phosphatase, nucleotidase. They break down non-transportable to transportable ions. There are some detoxifying agents like beta-lactamase, aminoglycosides, phosphorylases. They are responsible for drug resistance, osmoregulation, and this is important for osmosis due to presence of highly branched D-glucose. What are the clinical significance of capsule? It has got importance to the bacteria and importance to the human being also. They act as virulence factor by protecting bacteria from phagocytosis, helps in diagnosis by capsular swelling test, by immunological test, and acts as an agent for vaccine preparation. What are the different types of stains? Simple stain, if single stain is used like crystal violet, methylene blue. Differential stain, they bind to some types of bacteria but do not others, therefore providing a method of differentiating like gram staining, acid fast staining. And there are some special stain like negative staining, which stains the only capsule, endospore stain, flagellar stain, etc. And fluorescent dyes are also a special stain that can be used for mycobacteria. What is the basis of gram staining and ZN staining? The basis of gram staining, this staining has been discovered by Hans Christian Gram in 1884. The gram stain depends on ability of certain bacteria, that is gram product, to retain the complex of crystal violet and iodine after a brief wash with alcohol and acetone. But gram-negative bacteria do not retain the di-iodine complex and become translucent, and they can be counterstained with a safranin or red dye. Thus, gram-positive bacteria look purple under microscope and gram-negative look red. What is the basis of ZN staining? Basis of ZN is also structure in the cell wall. In addition to peptidoglycan and arabinogalactan, there is mycolic acid. This hydrophobic structure of microbe resistant to many harsh chemicals, including greater than and strong acids. If the dye is introduced into cells by brief heating or treatment with detergents, it cannot be removed by dilute acids. So they are known as acid-fast bacteria. So gram staining So this is gram positive, this is gram negative, and this is due to peptidoglycan layer. And the peptidoglycan layer of gram negative bacteria is very thin, only one. What information you can get from gram stain smear, or what is the clinical significance of gram stain smear? It helps in diagnosis of many diseases. It helps in identification of bacteria. If stained from colony of culture, it helps in guideline of treatment. Say how it helps in diagnosis of disease. Say if it is there stained from sterile body site, the presence of the bacteria with its morphology and arrangement, we can definitely diagnose. So for example, classical example is gonorrhea from urethral or prostatic smear. You can cure it. And bacterial meningitis from CSF. When you stain it from a colony, say a colony seems like maybe gram positive or gram negative, but if it is stained from colony, you can identify it. Say if it is gram positive cocci arranged in cluster, you can say it is Staphylococcus. If you cannot identify, but if you can differentiate between gram positive and gram negative bacteria, cocci and bacilli, it can guide you for treatment. What is the principle of diagnosis of microbial disease? We discussed it in another lecture. First of all, microscopy. It is commonly practiced for parasitic and fungal disease, also many bacterial disease. Second one is culture, commonly known. There is isolation and identification of positive agent by artificial culture, commonly practiced for bacterial disease. Detection of antibody or antigen from blood or other body fluids, practiced for viral disease, some bacterial and parasitic disease, and detection of nucleic acid, most popularly nowadays known as PCR. So what are the steps involved? You need some specimen from appropriate site. Then you will have to decide which type of microscopy you will do, like microscopy, diagram, or unstain preparation or stain preparation. Then if it is bacterial disease or fungal disease, you can go for culture. And if it is for virus, then cell culture can be done. Immunological test, very important, detection of antigen or antibody, and nowadays nucleic acid based technique, PCR. So best wishes for all students. Our next review class will be on bacteriology, if Almighty allows. Thank you very much. Thank you for your patience hearing. Assalamu alaikum wa rahmatullah.